Origin of cells and unicellular evolution. The origin of life is considered as the result of a long series of physiochemical changes which brought about by the first by chemical evolution, then by the biological evolution. The evolution up to formation of coacervate is termed as a chemical evolution, and from coacervates to simple cell structure it is called biological evolution. The origin of basic biological molecules. The earth has formed about 4.5 billion years ago, and the first evidence of life was more than 3.5 billion years ago, but the chemical evidence of life from as far back as 3.85 billion years ago. The first atmosphere was made up of hot hydrogen gas, too light to be held by Earth's gravity. The water vapor, carbon monoxide, carbon dioxide, nitrogen and methane replaced the hydrogen atmosphere. As Earth cooled, the water vapor, monoxide, carbon dioxide, nitrogen and methane replaced the hydrogen atom. As the earth cooled, the water vapor condenses and torrential rain filled up its basins, thereby forming the seas. In the first stage of chemical evolution, the molecules in the primitive environment formed simple organic substances such as amino acids. The ultraviolet radiation from the sun provides the energy for the transformation of these substances into the organic molecules. Further, the chemical evolution occurs in 4 stages. 1. Abiotic synthesis and accumulation of monomers or small organic molecules that are the building blocks of more complex molecules. 2. Joining of monomers into polymers, example protein and nucleic acid. 3. Formation of protobiont droplets which formed from aggregates of abiotically produced molecules and which differed chemically from their surroundings. 4. The origin of heredity during or before protobiont appearance. The discovery that RNA molecules can act as a catalyst in their own formation suggests that the RNA or a similar molecules may have been the first gene and the first catalyst. The concentration of a self-replicating RNA molecules would increase exponentially as one molecule formed two formed 4 and so on. In the competition for nucleotide, the most efficient of the self-replicating sequences would win and less efficient replicator would fade from the population. The steps of evolution. Free atoms. In presence of hydrogen, carbon and nitrogen it forms inorganic molecules, and in presence of hydrogen, water, methane, NH3, carbon dioxide and hydrogen cyanide it forms simple organic molecules. In presence of sugar, simple sugar, amino acids, fatty acids, glycerol, purines and pyrimidines it forms a complex organic molecules. In presence of polysaccharides, nucleic acid, lipids and proteins it forms coacervates or microsphere. In presence of a cluster of complex organic molecules bounded by fatty acid membrane and divided by budding it forms a pre-cell protobiont or protocell. In presence of nucleic acid, controlled coacervates, slow intake and outlet of components 3,800 million years ago, it forms a free polynucleotides. In presence of self-replicating nucleoproteins, it forms a first primitive cells. In presence of anaerobic prokaryotes, chemoheterotropic absorbs inorganic and divide to increase then number. To form a chemoautotropic cells. In presence of anaerobic prokaryotic chemoautotropic examples iron bacteria, sulfide reducing bacteria, nitrifying bacteria, they use sunlight for synthesis of food to form a photoautotropic cells. In presence of anaerobic prokaryotic photoautotropic, they originated due to the deficiency of inorganic compounds. They use sunlight to synthesize food to form an oxygenic. They did not use water as a raw material in photosynthesis. 3,300 to 3,500 million years ago, the oxygenic cell used water for photosynthesis and evolved oxygen gas. In presence of aerobic prokaryotic photoautotropic cyanobacterium procambarian, example orche spheroid, barbarosis to form eukaryotes organisms. In presence of multicellular organisms evolved due to the endobiosis 1600 million years ago. Then simple land plants are formed. The division of function between the DNA genetic information storage and protein catalysis was, according to the RNA world hypothesis, the new variant of self-replicating RNA molecules developed where the additional ability to catalyze the condensation of amino acids into peptides. The peptides thus formed would reinforce the self-replicating ability of RNA, generating even more efficient self-replicating system. They sometimes after the evolution of this primitive protein synthesizing system, the DNA molecules with sequence complementary to the self-replicating RNA molecules took over the function of converting the genetic information, and RNA molecules evolved to play a role in the protein synthesis. The protein proved to be the versatile catalyst and over time take over the function. The lipid-like molecules in the primordial soup formed a relatively impermeable layer around the self-replicating collections of molecules. The concentration or proteins and nucleic acids within these lipid enclosure favors the molecular interaction requires in self-replication. Abiotic synthesis of organic monomers and polymers. The various laboratory replications of early Earth, even with of a low concentration of oxygen, have been able to produce all 20 amino acids, sugar, lipids, nitrogenous bases, and even ATP. The abiotic synthesis and accumulation of organic monomers could have been a natural process on primeval Earth. The abiotic synthesis of polymers may have occurred when dissolved organic monomers splashes onto hot rocks. The fox has created polypeptide he called proteinoid by dripping dilute solutions of organic monomers onto solid sand or rock in the laboratory. The clay may have been an important substratum for polymerization because of its ability to concentrate organic monomers, which binds to charged particles of clay. Metal atoms such as iron and zinc at some of the binding sites may have functioned as catalyst, facilitating the dehydration reactions that linked monomers. The iron pyrates have also been proposed as a substratum for organic synthesis. The protobiont aggregates of abiotically produced organic molecules probably preceded living cells. The protobiont may have developed such capabilities as metabolism, excitability, and the ability to maintain an internal chemical environment different from their surroundings. Concept of operin and haldane. In 1922, the biochemist Alexander Operon proposed a theory for the origin of life early in the history of Earth, postulating that the atmosphere was very different from that of today, rich in methane, ammonia, and water, and essentially devoid of oxygen. It was a reducing atmosphere in contrast to the oxidizing environment of our era. In operant theory, the heat energy from volcano causes ammonia, methane, water vapor, and other components of the primitive atmosphere to react, forming a simple organic compounds. These compounds then dissolved in the ancient seas, which over many millennia becomes enriched with large varieties of simple organic substances. In this, the warm solutions, the primordial soup, some organic molecules had a great tendency that others to associate into a larger complexes to form a membrane and catalyzed enzymes which came together to become a precursor of earliest cells. Thus, Operon postulated that the first living cell arises spontaneously from this warm concentrate solutions of organic compounds compounds. This view was incidentally forwarded by Halden in England. Their view was attended by much controversy and remains untested speculation for over many years until a surprising experiment was conducted by Stanley Miller and Harold Urey using a simple equipment. The experiment of Miller in 1953. In 1950s, Stanley Miller and Harold Urey set up an experimental primitive atmosphere containing hydrogen atoms, ammonia, methane gas, and water vapor. They pass a spark through these gases to simulate lightning, then cooled to the system so that the gas would condense and collect in a watery solution. Within days, the system contained numerous complex molecules, including amino acids, purines and pyrimidines, some of the building blocks of life. The Miller-Urey experiment simulated the possible atmospheric conditions on primitive Earth to obtain some of the molecular building blocks of biological systems. The results of a Miller-Urey experiment have undergone several such refinement. 1. In living organisms, many molecules have the L configurations, but the amino acid formation in the Miller-Urey experiment were a mixture of D and L forms. The recent experiment shows that the natural processes could have selected the L amino acids from the mixture. 2. There is abundant evidence of major volcanic eruption 4 billion years ago that releases carbon dioxide, nitrogen, hydrogen sulfide, and sulfur dioxide. The prebiotic chemistry experiment using these molecules in addition to the ones in its original soup have led to more diverse molecules. 3. The long polymer had to be formed from simpler building blocks called monomers. 4. The Miller and Urey as well as others suggested that the life originated in hot pools at the edge of oceans. In whatever way the earliest stage of chemical evolution occurs, they resulted in emergence of monomers and polymers have probably remains unchanged in their general structure and function for 3.8 billion years. The first cell, the earliest cell that arises in the rich mixture of organic compounds were almost certainly chemoheterotrophs. The organic compounds that recurs were originally synthesized from compounds of the early atmosphere of carbon monoxide, carbon dioxide, nitrogen, methane, and such by the non-biological actions of volcanic heat and lightning. The early heterotrophs generally acquires the ability to drive energy from oceans in the environment and to use their energy to synthesize more of their own precursor molecules, thereby becoming less dependent on outside sources. The chemoheterotrophs causes oxidize organic compounds to carbon dioxide by passing electrons not to oxygen but to acceptors such as SO4 yielding H2S as the product. Formation of quiacervates. The large organic molecules synthesized abiotically on the primitive Earth were the precursors of colloidal aggregate. These colloidal particles were called quiacervates of oparin and microsphere by Sidney Fox. When oppositely charged colloids mixed, quiacervates were formed. These quiacervates droplets represents the old system that keeps accumulating materials from the environment. In quiacervates, the lipid molecules were joined end to end, forming a layer around each aggregate, which represents a simple lipid membrane. More complex microsphere and quiacervate functions as a pre-cell or pre-biont. Evolution of prokaryotes. A very significant evolutionary event in prokaryotes was the development of pigment capable of capturing the energy of light from sun, which could be used to reduce or fix carbon dioxide to form more complex organic compounds. The original electron donor for these photosynthetic processes was probably H2S, yielding the elemental sulfur or sulfate SO4 as the byproduct, but a lateral cell develops the enzymatic capacity to use water as electron donor in the photosynthetic reaction, eliminating oxygen as a waste. The cyanobacteria are the modern descendant of these early photosynthetic oxygen producers. With the rise of oxygen producing photosynthetic bacteria, the atmosphere becomes progressively richer in oxygen, a powerful oxidant and deadly poisonous to anaerobes. Origin of eukaryotic cells. Three major events must be occurred as a prokaryote give rise to eukaryote. First, as a cell acquires more DNA and fold it compactly into discrete complexes with specific protein. For this, a specialized protein was required to stabilize the folded DNA and to pull the resulting DNA complex protein complexes chromosomes apart during the cell division. The second, as the cell becomes larger, a system of intracellular membrane and a double membrane segregate the nuclear processes of RNA synthesis on a DNA template from the cytoplasmic process of protein synthesis on ribosome. Finally, the early eukaryotic process of protein synthesis on ribosome. Finally, the early prokaryotic cells, which are incapable of photosynthesis or aerobic metabolism, enveloped aerobic bacteria or photosynthetic bacteria to form symbiotic association. Some aerobic bacteria evolved into the mitochondria of modern eukaryote, and some photosynthetic cyanobacterium became the plasmid, such as the chloroplast of green algae, the likely ancestor of modern plant cells. Evolution of unicellular eukaryotes. The unicellular organisms at later stage of evolution found it advantageous to cluster together and acquiring more motility efficiently or reproductive successes than their free life single-celled competitor. Further evolution of such clustered organisms led to permanent association among the individual cells and eventually to specialization within the colony to cellular differentiation. The advantages of cellular specialization leads to the evolution of ever more complex and highly differentiated organisms. Many modern multicellular organisms could contain hundreds of different cell types, each specialized for some functions that supports the entire organisms. Anaerobes metabolism, photosynthetic and aerobic metabolism. All major metabolic pathway evolved in the prokaryotic before these were eukaryotes, probably within the first billion years of life on Earth. Various hypotheses was based on geological, molecular and metabolic evidence. Many millions of years for Earth to be developed an oxygenated atmosphere. Only a single-celled prokaryotes lived on Earth for more than 2 billion years, about 1.5 BYA. The atmospheric oxygen concentration becomes high enough for larger eukaryotic cells to flourish. The further increase in atmospheric oxygen concentration in the late Precambrian enables several groups of multicellular organisms to evolve. Anaerobic metabolism. The anaerobic metabolism is not efficient as aerobic metabolism. Molecules of glucose can only produce these 3 ATP molecules under anaerobic conditions, while it produces 39 with aerobic metabolism. ATP is what fuels the muscles. The anaerobic metabolism can only use glucose and glycogens, while aerobic metabolism can also break down fats and proteins. The intense bout of exercise in the anaerobic zone and in the red line zone with heart rate over 85% of your maximum heart rate will result in the using the anaerobic metabolism to fuel the muscles. Origin of photosynthesis. The light absorbing pigments may have protected cells from excess light and then becomes coupled with the electron transport system to drive ATP synthesis. A simple mechanism of photophosphorylation have been found in some archaebacteria called extreme halophils, in which the bacteriorhodopsin, a membrane-bound pigment molecule, uses the light energy to pump protons out of the cell. The resulting gradient is used for ATP synthesis. The development of pigment and photosystem co-optioning the component of electron transport chain may have allowed some prokaryotes to use the light energy to drive the electrons from H2S to NADP plus, and thus generating the reducing power to fix quantum reaction makes their own organic molecules. The nutrition of the green and purple sulfur bacteria probably is most like that of natural photosynthetic prokaryotes. Aerobic metabolism. In the aerobic metabolic processes, the human body uses a molecules of glucose to produce 36 adenosine triphosphate ATP molecules. The ATP is what fuel your muscles. The anaerobic metabolism, which is used for vigorous muscle contraction, only produces 2 ATP molecules per glucose molecules. So it is much less efficient. The aerobic molecules is part of cellular respiration and involves your cell making energy through glycolysis, the citric acid cycle, and electron transport or oxidative phosphorylation.